At Collaborations Pharmaceuticals, we use real drug discovery intelligence alongside artificial intelligence to develop clinical candidates for rare, neglected, and unmet therapeutic needs. Our core expertise extends to high-quality data curation and machine learning model building, which we offer as a service. We can apply our technologies to drug discovery and ADME tox predictions. We can also conduct in vitro testing in our own labs and coordinate in vivo testing through our collaborator network. We have utilized drug repurposing to get us quickly to promising molecules, as we have demonstrated for Ebola, Chagas, and Alzheimer's disease. And we can apply these approaches to virtually any disease. We are unique in that we publish our own results in peer-reviewed journals and share our data models, unlike other artificial intelligence companies. Please get in touch to learn more about how we can help you at collaborationspharma.com.
Hello, everyone. I'm Sean Eakins, CEO and founder of Collaborations Pharmaceuticals. Thank you for joining me today for my webinar, Molecules Designed by AI, the Commoditization of Machine Learning. It's great to see so many of you joining me today. I'm sure that you're looking forward to learning about the topic today, but I'd just like to share a few things with you before we begin. I obviously will try to show you how committed we are with using machine learning within our company, but I would also like to try to keep the information at a pretty high level for you. So I'm happy to answer any questions, and if you could please share those questions with us through the system that we have here. You can use the icon at the top of your screen to ask questions at any time through the presentation. You can also use the attendee chat functions on the left-hand side of the screen. If you have anything that you would like to discuss confidentially, just please make a note of that. And I look forward to answering any of the questions that you may have. I will also try to keep the presentation to about 25 to 30 minutes because I obviously value your time. And let's hope that this works technologically. This is the first webinar we're giving this year. And so without further ado, I'm going to dive straight into it. Obviously, you're seeing a lot of visibility around AI in terms of the application in the pharmaceutical space in the news. And definitely over the last month or so, this has increased as well. There have been many articles that have come out. Here are just a sample of those, really espousing the impact that AI is potentially having or is already having in pharma. Certainly, many of them have also commented on the growth in the number of companies that are using AI and applying it in the healthcare space. So there's really a lot more visibility about AI, a lot of buzz, and some of this, may be justified, some of it may not. The other thing I think that's also really highlighted in this particular slide, AI is everywhere in the industry. It covers all different facets from early stage, preclinical, through to clinical stage. So the application space is massive. There's obviously literally hundreds of companies focused on using AI now in the healthcare arena and drug discovery. And with that comes a very large number of investors. And I believe that on the order of literally billions of dollars have been pushed into AI companies and developing it. And certainly there are many big pharma that have been embracing it also. You've seen that with the increasing number of deals that have been happening recently. But there's obviously still, I think, a lot more that's going to happen in this space. And we're literally just at the beginning of this visibility. So AI is everywhere. And our particular interest in AI is really around molecules designed by AI. And to some extent, this isn't at all really restricted to the pharmaceutical space. Obviously, we're interested in designing molecules that have a therapeutic outcome, but the same types of approaches of design, make, test that are shown on this slide just as applicable to consumer products, agrochemicals, and animal health. So I think that although much of the focus so far has been on AI in healthcare and pharmaceutical space, we should also be thinking about these areas too, which also require, in many cases, small molecules. And clearly, if we can design, make, and test molecules more efficiently using machine learning, we can certainly also find molecules faster and test them. And that's really applicable if we're thinking about repurposing molecules. So the types of areas, obviously, where I think the technologies have already shown some benefit is in molecule design, but also in toxicology prediction, drug-drug interaction, etc. This is also getting to, I think, the idea of the evolution of the laboratory. So over the past couple of 100 years, we've literally moved from the laboratory being in the scientist's house to being an industrial laboratory with a whole organization that we pretty much take for granted today. And for the last 20 or so years, my laboratory just happens to have been the computer screen. So all of the experiments pretty much I've been doing have been on the computer, using models, using machine learning, etc. So we could also start to think about what will the laboratory of the future look like, specifically in the chemistry, molecule design space. And will the laboratory even exist to an extent? Will there just be literally boxes that make the molecules? And so I'll have a little bit of discussion about that as well today. How machine learning and artificial intelligence is really getting us to this point where it's changing the laboratory. Another thing we should be aware of is that AI didn't just magically appear over the last couple of years. It's literally decades old. And some of the work that we've been doing in the past, maybe over the last 20 or so years, is really just essentially being rebranded in some ways. And when we think of quantitative structure-activity relationships, which is what I started out doing back in the 1990s, now this has really been superseded by machine learning, primarily because we have massive data sets and much more powerful technologies, many more molecular descriptors, etc. And similarly, in the areas of systems biology from 15, 20 years ago, which was really when that particular area was just really taking off and becoming popular, those data sets were only starting to be created in the 2000s. And now we think of the massive amounts of biological chemical information, and we create knowledge graphs. And so that's kind of the hot term for this area. But really, I think there's a lot of rebranding. So what's old is really now new again. So it's important to take that into account. I think we're also at the stage where machine learning is a technology that's enabling us to build an assembly line of models. And then we use those models to create value. And I think this could be almost analogous to what Henry Ford did in creating the assembly line to make automobiles. And so what we maybe should think about is the stage that machine learning is at. And so we are essentially now creating that production line. So I think we're still at a pretty early stage, but machine learning is really now commoditized. Within our company, we've focused on putting together a number of core technologies, and we've given them all names, obviously. I'm not going to spend too much time on this, but the main technology for us is our machine learning tools, which we've called Assay Central. And this is our core technology for curation of models, data sets, and validation of models. We've also been able to, with this technology, build different types of models in different areas, and then we've bundled those together and created additional products. And so we've been able to create Megatox, which is essentially our toxicology platform, collections of human transporter models, which we've called Megatrends, and a collection of literally thousands of models built from data sets from Kemble, which would be useful for repurposing. And this product we've called MegaPredict. And I'm happy to share more information on that or answer any questions. But for the moment, I'm going to just focus a little bit on Assay Central. This is our main technology for curating data sets and then also building models. We now have 13 different algorithms, both classification and regression. This is obviously a moving target in terms of, there are many more algorithms out there, and so we're literally just adding to the collection. So we can build models with a data set across all of these technologies at the same time. We can look at many different types of different descriptors. We found the ECFP6 descriptor is particularly powerful, but we have access now to several different types of descriptors in this tool. And then we have options for different bit folding and types of cross-validation. And this is just one example of the output where we can show all the machine learning models on the left-hand side and really the metrics for each of the models along the top. So this is our workhorse technology, and we've been able to apply it in many different areas. Obviously, what's important to you is probably how the models are validated. And over the years, we've been very fortunate with our own lab and also with collaborators to validate the machine learning approaches in different areas. Obviously, because of lack of time today, I'm just going to focus on one area. But this slide really gives you an idea of some of the main disease targets that we've looked at, where we've done actual prospective prediction and to identify molecules with activity that weren't previously known. And we've put a lot of work into Ebola, and this primarily came from an initial project where we tried to curate a published data set of under 900 molecules. We built a Bayesian model and then used that model to score a library of drug molecules. And then out of that, we essentially just picked 3 molecules, tellurone, quinacrine, and pyranidine, and then tested them with the laboratory. And obviously, that's not something we could do ourselves in the lab, so we had to work with collaborators that had access to BSL-4, and all of these molecules were found to be nanomolar active. And then over the years, we've been able to subsequently perform in vivo testing in the mouse model using the mouse-adapted Ebola virus. And all 3 molecules were shown to have statistically significant survival. So this is one of the few examples, I think, where people have used machine learning, validated it in vitro, and then also in vivo. So we have a really nice complete story there. Whereas many of the examples, we've got a focus really on just the in vitro validation. We have now several examples where this has happened, where we've been able to take it all the way through to in vivo, both for Ebola and other diseases. Wouldn't it be wonderful if we could do this for thousands of targets or diseases simultaneously? And one way to really get to that point, I think, is to really capture all of the public data. And so most recently, we published an article where we used over 5,000 data sets from the Kemble database. We looked for data sets where there were over 100 compounds at least in each data set. So this covers all types of targets, all kinds of diseases, all different types of endpoints. And we compared multiple different machine learning algorithms. So we looked at 7 algorithms in total. And we didn't have to use like Amazon Web Services or anything. We have our own in-house servers. And obviously now the compute power that's available to a small company like ours, is something that would essentially in the past have been on a large mainframe. But now we can do this literally on our desktops. And so we looked at 5-fold cross-validation and also performed various external prospective predictions across different data sets. And the summary of this massive comparison is really shown on this slide. And I obviously would highlight the publication. If you want to take a look at that, we'd be happy to share that with you as well. But the outcome of this was looking across all the different types of machine learning models, Assay Central, support vector machines, etc., deep learning. We basically found that the Bayesian approach was comparable to support vector machines, deep learning, k-nearest neighbors, random forests, across all the different metrics and across all the thousands of different data sets when we look at either the rank normalized score or the difference in the rank normalized score. So this is important, I think, because it really pushes you away from thinking, one algorithm is literally going to be the best. Actually, looking at many algorithms, you may see that they're essentially comparable when you look across very large data sets. The other interesting point about this is that now we have this capability to have access to thousands of models, so we could use them for making predictions. And this will be useful, obviously, in a drug repurposing scenario where you may have one or several molecules and you want to figure out, well, what's other uses for these molecules. So we could take them as an input, run them through the thousands of models, and then look at the prediction scores that come out of the different models, and then look at the ranking of those scores and try to identify, which ones might be the best ones to test in vitro. This is definitely really what we're trying to get at with the MegaPredict application. And so now, obviously, with these massive data sets that are available in the public domain, for example, in Kemble, we can literally do that. I mean, this is a reality that is really available. We've also been focusing heavily over the last year or so on building up our toxicity model platform, which is MegaTox. And we've created hundreds of toxicity models. We've looked at different data sets coming from EPA, Tox21, ToxCast, specifically in the endocrine disruption area. We've put a lot of effort into that, and most recently published 3 papers looking at AR, ER, and aromatase. We've also published on modeling actual rat acute oral toxicity data for quite large data sets. That paper was recently published. And then also we've looked at predicting drug-induced liver injury, which is obviously very relevant for the pharmaceutical domain. MegaTox is an area that we're putting a lot of effort into. We're looking currently at modeling the properties that are shown on this slide, and these may be of interest to other areas outside of the pharmaceutical industry, specifically the consumer product and also the agrochemical areas. Just as an example, I'm going to show you one of these in the area of biodegradation today. There's a lot of data obviously out there for many of these different types of toxicity properties, but for biodegradation, we've been able to find 2 data sets in the OPERA suite from the EPA. And one of these has nearly 1,200 molecules in the training set, and then 400 compounds in the test set. So we've been able to look at this data set as an example with our Assay Central software. And the output of 6 of these machine learning algorithms shown here with the various different metrics also shown on this slide is essentially all of the models perform similarly. These are building classification models. We're finding the 5-fold cross-validation values are all greater than 0.7. The range is pretty narrow, between about 0.73 and 0.75. And then similarly, all of the other metrics are also comparable. And so this is quite interesting. We don't normally see this kind of limited variability across models. Normally you see 1 or 2 models doing really well and a few not doing so well. So this is quite a fascinating example. But again, it points to the potential impact of having multiple algorithms. Now, with this technology, we can also predict consensus across all of these models as well. So you get the best of all the different algorithms in one go. Just to shift direction slightly, obviously all of the previous slides that I've shown have really focused on the machine learning aspect of taking a data set, building a model, getting to that point where you can make predictions for existing molecules. Another area of interest to us that we've started to look at most recently is in the de novo design of molecules. So starting from scratch to literally produce molecules that haven't been produced before, and doing that all in the computer. This is an area that several groups have been looking at, and we're using recurrent neural networks to do this. And we can either take a series of molecules that we have information for and train this recurrent neural network to learn from that set, and then use public and or private data to focus that model to optimize various parameters. Or if we don't have a lot of information, we can use data from analogous targets, for example, and use reinforcement learning to try to come up with new molecule designs. This is obviously a very highly schematic version of what's going on, but we're using a long short-term memory RNN, and the molecules that come out of it, we're able to filter them with our synthesizability algorithms that we've been generating, and that at least gives us an idea that the molecules are feasible. And then we can work with collaborators to get their feedback on the designs and feed that back in. The advantage of this approach that we're using is actually that we're taking smiles as the string, as the input string. So it's producing pretty reasonable molecules from the outset. And the other thing, I guess, to take away from this is obviously the speed with which we can do this. The training and the learning is pretty fast, and the generation of molecules enables us to do multiple loops like this in a week. So this is a technology that we're excited to be applying in different areas. And one example that I'm going to show today is generating these types of algorithms for peptide design for GLP-1, which is a target, obviously, for diabetes. It's well known. The great thing about this type of approach using an RNN is that we could use any string of molecules. So, for example, using a string of using the peptide string of amino acids, and then training that with a library of peptides, optimizing it with physicochemical properties or even GLP-1 activity, anything that really tries to confine the molecule space that you're looking at, and then generating thousands or tens of thousands of examples of virtual peptides to then narrow them down to get to molecules of interest. And that could just be narrowing them down using a classifier model, for example. And so we've taken this idea with some adjustments to it. We've put a different kind of spin on this by thinking about learning from different types of peptides, not necessarily GLP-1 related peptides, but for example, antimicrobial peptides. And so we took a set of these antimicrobial peptide sequences, trained the RNN LSTM to generate then novel peptides that weren't in the starting set. And then we used our GLP-1 receptor activity model to score the resulting peptides. And interestingly, there were several that scored really well. And what I'm showing here is just one sequence. And that molecule is shown here. So this is a de novo designed peptide. This one is 29 amino acids long. It has a good predicted activity score. Our threshold for activity was 100 nanomole. And we get a really high applicability score. So that suggests that this is a reasonable prediction. And then the similarity using Tanimoto's similarity to the human GLP-1 ligand is about 70%. So this is a feasible looking molecule. And so we're actually currently looking at making this, and then we'll do the in vitro validation of it. But this is really just to show you one application of how we can use this de novo design approach. But theoretically, it could be applicable to any other types of molecules, of large or small in structure, as long as we have a string as the input to generate the actual molecules themselves. Obviously, which areas we want to apply this to will really depend on the cost or the ease of validation. Some of these areas may be easier to make the molecules than others. For example, macrolactones may be more difficult, whereas if we can make a peptide for a few dollars, that may be more feasible. Another area that we've been applying machine learning to is really in where we have very large data sets. And one particular technology we've been looking at is quantum machine learning. And this is something that we've recently published a preprint on. And our interest really here was obviously the what's known with classical computers. As you increase the training set size for support vector machine and deep learning models, it takes a very, very long time to build the model. And so what we felt was that's literally going to hold us back as we get larger data sets. And so could we speed it up by using a quantum computer to build the models? And so we literally have done that experiment here. It's pretty straightforward. We looked at different sizes of training sets for the models. And as you can see, the classical computer shows a linear correlation with increasing time taken to build a model with increasing training set size. But the quantum computer, interestingly, literally plateaus very quickly. And so this suggests a potential quantum advantage clearly in compute time. This would obviously be obvious to most people, but I haven't seen people do this experiment with data that's relevant in our space, specifically in the machine learning modeling data sets for drug discovery or toxicology. So this, I think, is an interesting simulation. We've also been applying the real quantum computers, and so we've been applying a 53-qubit machine from the IBM Q-Hub, which we are able to access here at NC State. We've been looking at our TB data sets of nearly 19,000 molecules, and then building what are essentially deep learning models on the quantum computer. And comparing the models that we build on the quantum computer with the classical computer, we've been able to show a pretty significant speed up. Obviously, the trade-off for this faster speed is a little bit of accuracy in the models. But this, I think, is a real-world evaluation of using a quantum computer for the machine learning in the drug discovery type space. I think what's really just holding back the widespread, application of this technology is probably the domain expertise. And most of the people that are working on quantum machine learning are not in our space, looking at small molecules and using them to build these types of models. I think the other aspect is really access to data sets. Clearly, Chemical provides us a large number of data sets, and obviously other data sets are out there in other domains like EPA, PubChem, etc. And I think really the other key piece that's missing is knowledge of what's really needed in the molecule design space. But if we can perhaps collaborate more tightly with folks that are doing the quantum computing, I think that's where we'll see more benefits. So this was really our first foray into applying machine learning to this area. And clearly you could imagine that this will be the next space that will start to heat up, I think, on the pharmaceutical side. One final example that I'm going to talk a little bit about today came about through our work with a DARPA-funded project called Lambda with SRI. And the PI on this project is Dr. Peter Madrid. And the challenge here was, could we actually use machine learning to predict UV-Vis spectra? The gold standard technology that's used is time-dependent density functional theory calculations. And this type of quantum chemistry calculation is obviously expensive to compute, and then it also takes quite a while, and it requires very powerful computers to do this. We've used a long short-term memory approach, again, using ECFP6 descriptors to generate predictive models for UV-Vis spectra. And the table that I'm showing here just shows how, over time, we've literally progressed from relatively small training sets and holdout test sets to now where we have 690 molecules in the training set and 299 in the test set. And we're getting average RMSDs. Initially, we're around about 0.2. And our latest examples where we've been able to do this have actually brought this figure down even lower to RMSDs of about 0.13. So why would we be interested in predicting UV-Vis spectra? Clearly, the UV-Vis spectra has a lot of utility in molecule identification. It's obviously used widely from an analytical point of view, but there's also applications in phototoxicity prediction, for example. So we could apply this to that area, and that crosses all different types of industries, the drug industry, agrochemicals, etc. It's also of value if we're trying to design molecules for coatings, or even to come up with new ideas for dyes, for example. We could even work backwards from an input spectra or from an input spectra to come up with a new molecule as a dye, or we could go the other direction to try to predict the colors from a structure itself. So we think that this is really exciting application of machine learning. We haven't seen it done to this extent with UV-Vis spectra prediction. And most recently, we filed a provisional patent on this application. So all of this, I hope, has given you a little bit of a flavor of how we're using machine learning and some of the areas that we're quite excited to be applying it to. If I was to put on, I guess, my crystal ball, and take a look into the future, I was thinking, what will it be like in 25 years? Obviously, I've been working in this space now for 25 years. What will it look in another 25 years? And certainly, the thing that I think is probably the clearest is that we're going to have all of the published data curated and modeled. I think we're already at that point where we can get most of it. But we'll literally not have to think about having to go out and do that. It will already be done, and the models will exist, and this will enable us to make then predictions, and this will be transparent. I don't think we'll even need to go to the extent that we do now to go through all of these steps to use technologies to build the models and make the predictions. I think it's going to be much clearer to do that. I think also we can think about the amount of screening that we need to do in the future. Certainly there will be, because we have the computational technologies to make the predictions, I don't think we'll have to do as much screening, and I don't think we'll need as much high throughput screening. We'll literally just be focusing on picking and making the right molecule or a few molecules. And I think we'll also have an idea of the endpoint before we even make the molecule. So we literally have predicted everything. So in the drug space, that will be, having an idea of the probability of success, e.g. FDA approval, before we even synthesize the molecule. That's definitely not something that we're thinking about now. I think that's something that will come. I mean, there's plenty of data that could allow us to make that prediction. And I just don't think it's been done. And so all of these models will be available and accessed, but I think it will be done in a different way. The end point of this is clearly that we'll change the footprint of the laboratory. The laboratory now will literally just be a box that will sit on our desk, assuming that we have desks. And efforts like the DARPA project that we've been involved in, there are multiple laboratories that are driving to this same goal where they integrate machine learning with automation. So essentially then the molecule design is autonomous. That box is the brain that's thinking about optimizing the molecule with the properties and making the molecule and doing the analytical. And that's where I'm obviously very excited to be a very, very small piece of that project. But I think the machine learning is the key piece that's going to drive that forwards. So that's the future, or at least my vision of the future. But I think the other thing that maybe is clear from this presentation is that you don't have to be a big pharma to use or benefit from artificial intelligence. We've obviously shown that machine learning is valuable for us as a company. And so I'd like to take a few moments really to end the presentation to show you what we've been doing at the company. But for us, I think machine learning is a valuable commodity. And this is certainly something we've been able to put out there in a number of publications to date. So just to give you a little bit of an idea about who we are as a company, essentially used machine learning and our efforts in this space to found the company in 2015. We're located in Raleigh, North Carolina, and we have 3 labs. And so we have the capability now where we've grown from just being a lab focused on machine learning to now having a lab where we can actually do our testing as well. We've been able to do this through the funding obtained through grants from the NIH and the DoD. And this has enabled us now to build a robust molecule pipeline, as well as provide services for other companies. So we are a stable partner for these types of collaborations. We've also been able to file a number of patents and provisional patents on new molecule IP and also repurposing of molecules. And over these years, we've been able to obtain 9 orphan drug designations on rare and neglected diseases from the FDA, as well as 3 pediatric rare disease designations. And over the last year, we've been able to obtain 6 trademarks on our software. So what do we do? I think you probably have a really good idea of what we do from my previous presentation slides. We're using the intelligence that we've obtained over the years of working in drug discovery and toxicology, model prediction, etc., and applying that alongside the machine learning, artificial intelligence approaches. And in the company, our focus is really on coming up with molecules for rare, neglected, and unmet therapeutic needs. But we also use these technologies that we've developed not only internally and with collaborators, but also provide them as fee-for-service sort of collaborations and contracts. And our focus is heavily around the curation, building of machine learning models, generating predictions, and obviously performing experiments, either in-house or with collaborators. And we've put quite a bit of emphasis in the early drug discovery spaces, but obviously we've been able to work in other areas as well. And we've been able to provide technologies and make these available to collaborators. And this has allowed us to build up a pretty extensive pipeline. I'm not going to go into any detail here, but as you can see, machine learning has had a large role in many of these projects. My next webinar next month is going to focus in detail on the molecules in the pipeline. And I think the benefits of working with us include really our agility. We're a small company, but we're very flexible. So we're able to work on your challenges pretty quickly. We're able to turn things around pretty sharpish. For example, the peptide RNN example that I showed went from idea to concept in less than 3 days. We also have a very sustained productivity, and this is also seen by our number of publications, which from 2019 went from 22 papers to 2020, where we had about 29 papers, and majority of these are on machine learning. We're also very adept at obtaining intellectual property based on the number of patents and provisionals filed. And we have over 25 years of computational expertise in drug discovery and computational toxicology that we can put at your disposal. So just a final slide, really. We've worked with a number of companies to give you an idea of the types of things we've been able to do. We've worked on endocrine disruption. Previously, we've put a webinar out there on our endocrine disruption work. So I certainly would encourage you to take a look at that. That's available on our website. We've been able to put out multiple papers in this space, working with Collaborator, which is a multi-billion-dollar company. We have a long-term collaboration with them, which has taken multiple years, multiple projects. We've worked with a second consumer product company in a similar space. And we've also worked with a multi-billion-dollar rare disease company, designing new molecules for them and generating ADME predictions. And finally, we've been working with a small animal model CRO, predicting in vivo activity as well as in vitro off-target activities. None of this work really would be possible without funding from the NIH, DoD, and also the North Carolina Department of Commerce. And with that, I would like to thank all of my colleagues and collaborators that have enabled this work. And I would be happy to answer any questions. And if you haven't posted them yet, please put them in the audience question box shown here on the slide. If you would like to get in touch, feel free to email me at sean@collaborationspharma.com, and I'll be happy to answer them. So I'm now going to take a look at all of your questions, and I'll try to hopefully get through them as quickly as I can. Thank you all for attending. If you have to leave, we'll be recording this, so we'll be sharing the link with you later, and so you can take a look. And I'm just going to take a couple of minutes, hopefully, to try to answer these questions. Okay, so do you have any restrictions on the peptide length? It's a good question from Maria. Thank you, Maria. In the model that we built so far, yes, we restricted it, but in general, you could make that flexible, depending on, obviously, the area that you're working on. So we kind of put some restrictions on it for our particular project, obviously, because the number of peptides could be potentially infinite. So you obviously do want to put some constraints on it. Could you please comment, this is from Senthil, could you please comment on how descriptors, variables used in the models have changed, if any, over time? Do deep learning models give better predictive power overall, or are they specific to systems? That's a massive question. I would probably have to write a paper to address it fully, but it's a good one. Descriptors, we've definitely found that descriptors that have been widely used for probably a couple of decades, like FCFP6 and ECFP6, descriptors perform really well with the types of unbalanced data sets and various drug discovery, toxicology projects that we work on. So I tended to focus on those. Obviously, there are many, literally thousands of descriptors out there. There are also collections of tens of thousands of descriptors that people have made available, many of them obviously publicly available. So there's almost an infinite choice of molecular descriptors. In terms of deep learning, I've not really seen that much difference between deep learning and support vector machines and random forests. they all seem to be pretty much in the same space. We've published a number of comparisons showing that as well across different data sets. I have also another question on peptides. Is the peptide design also based on SMILES? In this particular model, no. We didn't use SMILES. We used the amino acid strings. But we could use SMILES if you wanted. That's definitely doable. You just any string that would represent the structure. But in this case, the actual model was trained, or it learned how to put amino acids together. Do the models consider biological background, e.g. blood, pH, or osmolarity when computing de novo outputs of peptides? In this case, which was a relatively simple example we put together, no, but they can be trained on other features. So if we had data for that, we could definitely use that as an input to train. There is a question about the ratio of actives and inactives in the machine learning and quantum machine learning models. And in this case, that obviously, for the quantum machine learning models where we had the same TB data set, we had different cutoffs. That obviously changed with the different threshold. And we have also experimented with automatic threshold cutoffs with the machine learning models. So the Campbell data sets, I believe that was where we used an automatic threshold to look at the cutoffs. So I definitely point you to that paper. And also for the preprint, for the quantum machine learning, I think that will give you an exact number of how many molecules are in the active versus the inactive for that molecule. So for that data set. Thank you for that question, Gustavo. Okay, UV-Vis, another question. How did you choose ECFP6? Have you tried shorter, longer radiuses? I think we've looked at several fingerprints. We've also looked at compression of fingerprints, but we focused on those initially because of obviously our efforts in the space. But we have been exploring other fingerprints, but so far the ECFP6 seem to work really well. Obviously, as we're getting the RMSDs down in that area, we're doing pretty well, I think. Okay, I've got some more questions here, which I'll try to get through quickly. How do you define the applicability domain of the developed models? We definitely have published on that with the early versions of Assay Central, so I definitely point you to the papers relating to that. Initially, I think we looked at the overlap with molecules in Campbell to get an idea. We've also looked at overlap of fingerprints and to get an idea of, do the molecules share fingerprints that are in the training set? So that actually is probably the best way to define that. Okay. All right, one question is therapeutic projects. This is from William. Therapeutic projects are each individual and unique wars. How do you determine in which projects and at what point in the project will models help? That's a great question. People generally would tend to come to us if they have a particular issue. So maybe they're stuck in an area where they want to try to get out of it. Like, for example, they need new ideas of molecules that could overcome a particular issue. There could be an ADME liability, for example, metabolic stability. In areas like the consumer product space, this is definitely more driven by regulatory, thinking about how could they replace some of the testing that they have to do, or how could they refine, focusing on molecules that they need to test. So definitely there's a lot more interest, I think, especially in the endocrine disruption and other tox endpoints from the consumer product space. That's where they would come in. So yeah, working between different industries, each has their own different needs. Obviously in the pharma space, yes, we're going to have potential liabilities. Can we optimize something in lead optimization versus can we use the machine learning models initially just to come up with hits for a particular project? Both very different. Also very different in terms of, do we want a local model versus a model maybe that is much broader? The types of projects maybe that we've worked on with industry, we've had a bit of a mix of both. William again asked the question, how do you incorporate the knowledge gained during project execution by computational chemists or experienced medicinal chemists? E.g., that there is a focus on centers of metabolism, or there is a need to definitely preserve certain specific interactions? That is a very good question. It would probably take me a while to answer that. I'll try to follow up with you afterwards and answer that if that's possible. I definitely think that the value of these types of technologies is enhanced if there is that feedback from medicinal chemists. We can definitely build that into the RNN types of approaches, I think, as well, where we try to focus on maybe where the molecules are changing. And certainly we could use these technologies also to create libraries of things like prodrugs. If you knew exactly where you wanted to modify the molecule, and we could, literally just say, this is the area we want to change, focus on making the analogs in that space. Murat asked the question, are 3D descriptors superior to 2D? We haven't really done big comparisons of it. I mean, we find that we get really good models already with 2D. So I don't know that 3D is going to give us much more. There's a lot more complexity, I think, to that as well, in obviously generating the conformations. So the cost benefit of doing 3D over 2D, it may not be worth it. I think other people have also done the same comparisons in the past as well. William again asked the question, do you imagine incorporating chemical knowledge or insights such as aromaticity, pKa, biofeaturization, or something like change density? I guess that should be charge density rather than fingerprints. I think there's probably, definitely a lot of flexibility here in what we could incorporate into the RNNs. I think we're still kind of at the early stages of really getting to grips with what they could do. But from what I'm seeing, there's a lot of potential. And also, how do we bring the technologies together with what we've built up from the purely machine learning side of things on data sets and public data? Putting that intersection together between the de novo design piece and the different spaces that we could play in there, and then how we bring the models is also, is ripe. I think there's a lot that could be done. Gabrielle asked a question. I think this will be the final answer I'm going to give because of time. Slide 26, vision of the future. You talk about finding molecules that could be approved easily by the FDA. Does that mean that for now it's hard to get a cross calculation to find a drug that has a good score for a target and low toxicity and biodegradation? Is it because of the limited amount of data? I guess the quick answer is, I think it's probably because these data sets are not put together. We kind of have silos of the data sets. So we have the silos in something like Chembl that focuses on the bioactivities. And then we've got silos of information that are in proprietary databases where people have looked at clinical success, clinical trial success. And we've got EPA databases that are looking at these more environmental type pieces of information. And I think if we're really trying to get at that whole end-to-end process that's done computationally, we really have to bring those together. So there's definitely, we've got to traverse the public versus the private information. Probably the public amounts of information that are out there on clinical success are relatively small data sets compared to what's in proprietary databases. So that would hold us back. But I think the real power is if you can tap into all of this information, and especially if you're looking at the drug space or you're trying to come up with molecules that have to get EPA approval, that's probably equally analogous. So that's what I think is probably holding us back from that future. But it's feasible that we could overcome it. So anyway, thank you all for the questions. If I haven't answered them now, we'll try to address them by emails to you individually. Thank you all for your attention today. And if you have any other questions, drop me a line at Sean@collaborationspharma.com, and watch out for the next webinar. Thank you all for your attention. Take care.